All right, viruses. So, what's distinguishable? You remember they are acellular, so they do not have cellular structures. They obligate intracellular parasites. And here's why. Viruses have no ribosomes, so they can't carry out protein synthesis independently. And they have, now I'm going to use the word metabolism, and I will explain myself. So here, under the term metabolism, we should understand viruses cannot convert energy. They can't produce ATP. You see what I'm saying? They can't generate ATP. They have to use cellular ATP. Am I clear? These are two reasons why viruses are obligate intracellular parasites. So structurally, they can be divided into two categories. One category of viruses are called, as you may remember, naked viruses. And another category is enveloped. Naked viruses consist of the capsid and the genome, while the enveloped viruses consist of capsid, the genome, and the outer envelope. All right, so far so good. So envelope is made up by viral proteins and phospholipids. You remember phospholipids are components of the cell membrane. So essentially a virus, when it leaves the cell, it steals parts of the cellular membranes, whether it's a cytoplasmic membrane or Golgi or endoplasmic reticulum. Capsids consists of the viral proteins, and just proteins, always proteins, and there are three main types of capsids. Capsid can be icosahedral. Now, if you like me and don't have well-developed 3D imagination, I can only tell you this. Imagine you have 20, what's the name of the triangle where all sides are the same size?
Equilateral.
Huh? Equilateral, okay. You have 20 equilateral triangles that are assembled in a sphere-like structure. That makes sense? For myself, it's not a right approximation, it's incorrect approximation, but it's like, like a soccer ball. If you look at the soccer ball, it's not really a sphere, it's individual polygons. Does that make sense? So icosahedral capsids are most common. For instance, coronavirus has icosahedral capsid. Helical. Pretty straightforward. Individual proteins are assembled into the helix, and this is what I like about science. One of the things that I like about science, but particularly about scientists. We're going to name two categories, then we're going to get real tired of classification. We just dump everything else into a category called complex capsids. Okay, so there's no common distinction here. Does that make sense? Everything that's not icosahedral or helical is considered a complex capsid. Now, what about genome? And that's an interesting conversation. Now, viruses are not cellular, so therefore, are they living? No. And so all the living things, as far as we know, contain double-stranded DNA genome. Well, viruses aren't living, so they don't have that restriction. They can have double-stranded DNA genome, like herpes viruses, They can have single-stranded DNA genome, like parvoviruses. They can have double-stranded RNA genome, like, for instance, rotaviruses. And they can have two types of single-stranded RNA genome, the positive and the negative. And I'm almost sure that you never encountered the term positive-stranded RNA or negative-stranded RNA. Okay, now, we're going to figure out what it means together. How about that? So, what is positive single-stranded RNA genome? You take this genome, hypothetically, and you put it into the cell. Are you following me so far? You with me?
This is positive.
Yes. So you take this positive single-stranded RNA genome, put it into the cell, and cellular ribosomes can translate it into a protein right away. Does that make sense? So for instance, if you take single-stranded RNA genome of yellow fever and you stick it into the cell, and by the way, you can do it, like it's doable. I did that with these hands. Okay? You stick it into the cell will start making proteins right away. If you do the same with negative single-stranded RNA genome, cell can't do it. Now, question 5 out of 10. Remember I told you about the complexity? So this is 5 out of 10 question, maybe 4 out of 10 question. What should happen to negative stranded RNA genome before proteins can be synthesized?
Turn it to positive.
Yes, absolutely right. You should turn it to positive. Now, the question. Say you have, let's draw it. Negative stranded RNA genome on, you should use it to make positive. Now, second half of that same question. How these two relate to each other? So you should use this one to produce this one. You're absolutely capable, like you have the knowledge to answer this question.
I think it's a template.
Huh?
It's like a template? Is that what you're...
A what?
A template?
What is the template? Go ahead. What is the template?
The single strand negative RNA.
Okay, template, and then you synthesize positive single-stranded by which principle?
Transcription.
You're right. It's basically transcription. It's not really transcription, but it's basically transcription. But how do they relate? I'm looking for one word that starts with C. Two strands. Huh? Two strands. Bingo. They complementary, right? So you take negative stranded and you take A, you put U. C, you put G. And you just, so they are complementary strands. Does that make sense? And then you can, translate single-stranded, positive single-stranded RNA. Now, here's an important feature. So, let me ask you this. And this is not, don't overthink my question now. This process, translation, when RNA is used as a template for protein synthesis, this process, can it happen in a human cell? Does it happen in a human cell? Yeah, it does. So you can literally take, so for instance for yellow fever, when it infects the cell, its RNA just goes into the human cell and gets translated right away. Does that make, you with me so far? Now, this process, synthesis of one RNA molecule using another RNA molecule as a template, does it happen in a human cell? Don't overthink.
Can you repeat that?
Yeah. Do human cells normally, regularly, synthesize one RNA molecule using another RNA as the template? Have you heard of it? No. It doesn't happen. It does not happen. So this process is unique to viruses. Does that make sense? This is why, if you look at all negative strand RNA viruses. They all, in the structure, have an enzyme that will run this process. They have to carry it with them. Are you following me? So for instance, coronavirus, which is negative stranded RNA virus, has the RNA-dependent RNA polymerase. In the virion, in the particle. Measles, mumps, influenza, they all have it. Because they all negative single-stranded viruses. Does that make sense? Questions about that part? Okay, a few words about the virus discovery, and then we're going to jump to the general concept of the virus infection. Viruses were discovered in very late 19th century. All I want you to know is two things. First of all, first virus that was discovered was virus of plants. Animals came much later. It was tobacco mosaic virus. Second thing, the discovery came after...
To know that name of the virus?
No, come on, no, just plant virus. Plant viruses were discovered first. Next, you need to know kind of an idea. So what happened? The first viruses that were discovered. How they were discovered? They were passing through the filter. Remember, you did filtration. So that filter contains bacteria. It stops bacteria. Well, it doesn't stop viruses from going through. And this is how they were discovered. They went through the filter, and that's why they were first called filtrable agents. Now, last little bit, just to give an idea how small they are and how many of them are there. You don't have to write it down. I just want you to have a deep appreciation of viruses. The estimated amount of viruses on Earth is sufficient to fill 2 football stadiums all the way up to the top, which is not really a lot. The estimated number of virus particles in the world is 10 in the power of 31, which is more than the number of stars in the whole known universe, which is insane. Okay? And most viruses are not pathogenic. In fact, you eat a lot of viruses, the plant viruses and insect viruses, every time you consume any fruits or vegetables. You eat like a ton of them. That make sense? Okay, moving on. So we're going to talk about 3 types of virus infection. We're going to start with bacteriophages. So bacteriophages infect only bacteria and archaea. They don't infect plants, they don't infect protozoa, they don't infect humans or fish or insects.
These are the types of
This
viruses.
is a very special class of viruses, and I want just to review the process of infection, of how bacteriophages do that. We basically did that. Look. So you've got several steps here. You start, and this color is going to represent whatever virus does. So you start with the bacteriophage, and this is the schematic. You have this wonderful image in the notes that shows you a bacteriophage like this. With a capsid that contains nucleic acid, the tube, the fibers, and the pins. This is just one specific class of bacteriophages. Does that make sense? They do look like alien spaceships. They use fibers to attach to the cell, and they use pins, which boggles my mind completely. They use pins to drill the hole in the cell and get the genome in. And this is a distinctive feature of bacteriophage infection. So first step is when they attach to the surface of the cell, is called, not surprisingly, attachment. Okay? And then during the second step, which is penetration, only viral genome enters the cell. Does that make sense? The genes in a viral genome get expressed during the third step called biosynthesis, which results in the production of multiple copies of viral genome and viral proteins. Proteins and genomes during the step number 4 assembly form particles. And then these particles leave the cell during 5th stage, which is egress or release or exit, call it whatever you want. All those terms are absolutely acceptable. So far makes sense?
What happens in biosynthesis?
Proteins and viral genomes are synthesized. Does that make sense? Good? Now, look. What happens to the cell as the result of this infection? What do you think? When they just start popping out of the cell? It dies, right? It dies. So, the situation when virus just slams into the cell, kills it, leaves, this is called lytic infection, and the bacteriophages are called lytic bacteriophages. Am I clear so far with lytic bacteriophages? Good? Good? Second option, it's nothing complicated. Second option, we also discussed it when we talked about transduction. Second option is you have a virus, bacteriophage, that introduces its DNA into the cell, then DNA gets incorporated into bacterial chromosome in the form of a prophage. Okay, here. Can you see that? Prophage. So every time the cell divides, prophage divides with it. Okay, now, big question. Can it get out? Yes, it can get out. Usually under stress, prophage gets removed, and then this steps, biosynthesis, assembly, and release ensue. The point is, cell doesn't die right after the infection. Am I clear? So these bacteriophages that don't kill the cell immediately, which genome gets incorporated into the chromosome, are called lysogenic or temperate bacteriophages. So far, does that make sense to you all? Okay, good? All right. Questions for bacteriophages? No? Okay. So when I say eukaryotic viruses, of course I don't mean that viruses are eukaryotes. I'm talking about the viruses that infect eukaryotic cells. Am I clear? So here we have a generic cell, and remember what we're talking about here is sort of a general rule. It's not something that happens all the time. There are slightly different strategies. But look, first, virus has to attach to the surface of the cell, and as you learned from our conversation about HIV, the first step, the attachment step, requires a receptor. A receptor is an absolute must. Now, very, very, very simple question. Cells that have a receptor, can they be infected?
Yes.
Yes. Cells that don't have receptor, can they be infected? No. Make sense, right? So cells that have the receptor, they have a special term for them. Those cells are called susceptible. Once the virus binds to the cell, it is taken in as a whole. Okay? So penetration two main mechanisms for penetration are fusion, This is when, look, here, when envelope, which consists of phospholipids, fuses with a membrane, okay, releasing the capsid into the cytoplasm, or endocytosis, that's very, very common. A vesicle packages the virus and takes it in. When the virus is in the cell, now look at the first difference between bacteriophages and eukaryotic viruses. It's not just the genome, it's the entire capsid that gets in. Sometimes an entire enveloped structure, okay? Then during the third step, uncoding, the viral envelope gets all mangled up. See? So basically undresses the virus. Does that make sense to you? So you see how it all like broken in my image. Okay? Now what happens to the viral genome after uncoding? Two main directions for viral genomes, and this is in general, there are multiple exceptions. If it's the DNA genome, it goes into the nucleus, okay? And if it's the RNA genome, it stays in a cytoplasm. Does that make sense? Now I have a question for you. Why does DNA want to go to the nucleus? Huh?
To be replicated.
Excellent. 1 point, to be replicated, because DNA is in the nucleus. And all the machinery for DNA replication that virus may as well lack, not have, is in the nucleus. It will just hijack the cellular machinery. What is the second reason, which is a little bit more subtle? Okay, where is cellular DNA? In the nucleus. Is it normal for DNA to be in the cytoplasm? No. So many DNA viruses basically hide from detection by, now you're familiar, pattern recognition receptors. There's a system called GAS-STING, which is basically a system of pattern recognition receptors that recognize DNA in the nucleus, in the cytoplasm. So if DNA is not in the cytoplasm, it's perfect, right? There are exceptions. Such DNA viruses as smallpox, for instance, they remain in the cytoplasm all the time. They don't care. RNA viruses, such as influenza, we're going to talk about it today, they, on that end, they replicate. They are RNA viruses, but they replicate in the nucleus. Makes sense so far, right? Now, I'm going to use RNA viruses as an example just for the brevity. So then you have step 4. Synthesis of viral proteins and viral genomes. Biosynthesis. Now it's important to understand, look, not every cell will support the biosynthesis pathway. Am I clear? Some cells, and it's kind of a mind-boggling idea maybe, but listen, some cells that have a receptor, some susceptible cells, virus can get in. But for one reason or another, it's not going to reproduce within the cell. Does that make sense to you? So cells that allow for intracellular biosynthesis are called permissive cells. Good example, using coronavirus as an example. Coronavirus binds to the receptor that is found on the cells of respiratory passages. Same exact molecule is found on endothelial cells in the blood vessels. Are you with me? Well, turns out we can easily detect the virus in the respiratory cells, but virus doesn't seem to grow in endothelial cells. So in the human body, it looks like respiratory cells are both susceptible and permissive, while endothelial cells are only susceptible. Virus can get in, and that's the end of the story. That makes sense? Clear? Okay. What happens afterwards? Nothing different from bacteriophages. Assembly. And egress. For the most part, when, look here, when naked viruses get out of the cell, usually the cell dies right away. Generally speaking, naked viruses kill cells faster. When enveloped viruses get out of the cell, they tend to kill them slower because they don't pierce the membrane, they just pop out of it, and the membrane has a chance to merge behind them. That makes sense to you? Good? You may wonder, what kind of, basically like a description of what's going on at the step, and what's the name of the step, like matching the terms, okay? Does that make sense? Good? Okay.